You probably know your blood type. Maybe you were told once at a doctor's appointment, or you checked it before donating blood. A positive, O negative, AB. A letter and a symbol. One of the most medically critical facts about your body, filed somewhere in the back of your mind and rarely thought about again. But almost no one asks the deeper question. Why does it exist at all? Not what it means for transfusions. Not which type is the universal donor. The real question, why does the human body flag its own blood cells with molecular markers in the first place? And why does that flag come in 4 different versions? Why aren't we all the same? Every red blood cell in your body, and you have roughly 25 trillion of them, is coated on the outside with a layer of molecular tags. Some of those tags are sugars, some are proteins. The specific combination your red blood cells carry, determined entirely by your DNA, is what we call your blood type. In the ABO system, the most clinically critical of the 48 blood group systems currently recognized by the International Society of Blood Transfusion, the tags come in 2 main varieties, the A antigen and the B antigen. Type A cells carry the A antigen. Type B cells carry the B antigen. Type AB carries both, and type O cells carry neither. Your immune system learns your own tags early in development, registers them as self, and treats anything that doesn't match as foreign, triggering an attack the moment incompatible blood enters your body. This is why blood transfusion went from being one of the most lethal procedures in medicine to one of the most routine. Before 1901, physicians transfused blood without understanding why some patients survived and others died. The mechanism was invisible, and the failure rate was devastating. That year, an Austrian physician named Karl Landsteiner, working at the Pathological Anatomical Institute of the University of Vienna, noticed something in a series of test tube experiments. When he mixed blood samples from different people, some combinations caused the red cells to clump together violently, others didn't. The pattern wasn't random, and it wasn't contamination. It was identity. Within a year, Landsteiner had mapped 3 types. A 4th, AB, was confirmed in 1902 by 2 of his colleagues. In 1907, just 6 years after Landsteiner's paper, a physician named Reuben Ottenberg at Mount Sinai Hospital in New York used blood typing to complete the first documented safe transfusion on record. Landsteiner received the Nobel Prize in Physiology or Medicine in 1930. He is still called the father of transfusion medicine. During World War I alone, his discovery saved tens of thousands of soldiers who would otherwise have bled out on the field. But the discovery answered one question and opened a far larger one. Blood types clearly mattered for transfusion compatibility. The immune system clearly read them as identity markers. What nobody could explain was why those markers varied at all, why the human body came in 4 incompatible versions rather than one unified standard. In 2012, geneticist Laure Segurel of the University of Chicago and her colleagues published a study in the Proceedings of the National Academy of Sciences that reframed the entire question. Segurel's team analyzed the blood type gene, specifically the enzyme it encodes, which determines what sugar gets attached to the surface of your red blood cells, across 40 different primate species: humans, chimpanzees, gorillas, orangutans, gibbons, macaques, baboons. What they found was unexpected. The A and B blood types in humans don't date back merely to the common ancestor of humans and chimpanzees, roughly 6 to 7 million years ago. They are far older. The divergence between the A and B versions of the gene traces back at least 20 million years to a common ancestor that predates the emergence of every modern primate species alive today. For context, 20 million years ago, there were no hominids, no recognizable monkeys in the Americas, and the mammals that would eventually become whales were still walking on land. Blood types predate the human genus by a factor of 3. What Segurel's team found was stranger than the age alone. When they compared the A version of the gene across species, they found that a human with type A blood is genetically more similar in that specific region of the genome to a gibbon with type A blood than they are to a human with type B blood. Your blood type connects you more closely to a primate in the rainforests of Southeast Asia than to the person sitting next to you. Right now, your 25 trillion red blood cells are carrying a molecular signature that has been maintained generation after generation through 20 million years of evolutionary history. For that signature to have survived, something must have been preserving it. Evolution doesn't maintain variation without a reason. Something, or several somethings, must have made each blood type worth keeping alive across every primate lineage, across every continent, across every extinction event the world has thrown at the primate order. But of every force in human evolutionary history that could explain why this variation persisted, one stands in a category by itself. Plasmodium falciparum, the most lethal species of malaria parasite, infects roughly 200 million people every year and kills hundreds of thousands of them. The vast majority are children under 5 in sub-Saharan Africa. Across the millennia of human evolution, before medicine, before mosquito nets, before any intervention, the death toll would have been staggering. Population geneticist Joshua Akey of Princeton University has called blood types a really strong target of positive selection throughout our history. Malaria is the clearest reason why. In 2007, a research team led by Jane Rowe at the University of Edinburgh conducted a case control study of 567 children in Mali, West Africa. Each case of severe malaria, defined as unrousable coma, severe anemia, neurological impairment, repeated seizures, or evidence of organ failure, was matched against children with uncomplicated infections from the same region. When sorted by blood type, the result was stark. Blood group O conferred a 66% reduction in the odds of developing severe, life-threatening malaria compared to all non-O blood groups. The odds ratio was 0.34. The p-value was below 0.0005. Children with type A or type B blood, infected by the same parasite in the same village, were dying at rates that children with type O blood were not. The question was why. In 2015, Professor Mats Wahlgren and his team at the Karolinska Institute in Stockholm published the mechanistic answer in Nature Medicine. They had spent years hunting through the variants of a protein family secreted by Plasmodium falciparum called rifins to find the specific variant driving the worst outcomes. When falciparum infects a red blood cell, it hijacks the cell's surface, deploying rifin like molecular glue. Infected cells become sticky, binding to uninfected cells and forming clusters called rosettes. Those rosettes lodge in the microvasculature of the brain and organs, blocking blood flow and starving tissue of oxygen. In severe malaria, it isn't the parasite that kills you, it's the clotting, and the rifin protein bonds aggressively to the A antigen on type A blood cells. On type O cells, which carry no A or B antigens, rifin can barely grip. The rosettes that form in type O blood are small and fragile. The rosettes in type A blood are large, dense, and lethal. 70% of all rifin variants belong to a subclass called A rifin, proteins specifically structured to exploit the A antigen. The parasite had, across its own evolutionary history, built a weapon tuned to one of the most common human blood types. And the human genome answered back over hundreds of thousands of years, quietly selecting, in the most malaria-burdened regions of the world, for the one blood type the weapon couldn't hold. The evidence is written on the map. In Nigeria, where 97% of the population lives in malaria-endemic zones, more than half the population carries blood type O. The same pattern holds across the Congo, Ethiopia, Ghana, and sub-Saharan Africa broadly. In Scandinavia, where malaria was historically rare, type A blood runs at 45 to 50% of the population in some countries. In Japan and Korea, type AB, rare almost everywhere else, reaches about 10%. In the indigenous populations of the Americas, peoples who migrated through Beringia and spread south through the continents, type O runs at extraordinary concentrations. In Peru, 71%. In Ecuador, 75%. These populations carried the O-heavy genetics of their founding ancestors, and the Americas had no malaria before European colonization, so that signature was never pushed back toward diversity. Each regional distribution is a fingerprint. The map of blood types around the world is not random. It is a record of every epidemic that was lethal enough to change who survived. And malaria is only the most visible example. Type O blood carries higher susceptibility to cholera and plague, diseases that historically struck hardest in dense, low-altitude, river-adjacent populations. Type A carries elevated risk for smallpox and is most common in the northern European populations, where cold climates suppressed mosquito-borne illness. Every blood type is partial armor against some threats and a measurable vulnerability to others. None of them is universally better. The diversity is the strategy itself. This is not a story that ended in the past. The WHO estimated in 2024 that malaria killed 597,000 people that year, more than 75% of them children under 5 in Africa. The evolutionary pressure that built the current distribution of blood types is still active. The arms race between Plasmodium falciparum and the sugar molecules on your red blood cells never stopped. It slowed in some places, accelerated in others, and is now accelerating again as drug resistance rises in malaria-endemic regions. And the map of blood types still shapes medicine in ways most people never think about. Type O blood is the universal donor, compatible with every A, B, O type, which means it is called first in every emergency trauma situation. In sub-Saharan Africa, where type O dominates the donor pool, supply is never enough. The same evolutionary pressure that made type O common in those populations also makes it perpetually scarce for emergency medicine across the entire region. Your blood type isn't just a fact about your body. Every time you give blood, or receive it, or sit in a hospital waiting for a transfusion match, you are inside a system that was shaped by the evolutionary history your ancestors survived. And the science is still catching up. In 2025, a study in Scientific Reports traced the blood types of early modern humans who lived across Europe and Asia between 46,000 and 16,500 years ago, alongside evidence from Neanderthal remains. The researchers identified the Persian Plateau as a critical genetic incubator, a region where migrating humans paused for roughly 15,000 years and diversified, developing new blood type variants before spreading into Eurasia. The same effect played out in Beringia before the Americas were populated. Blood types weren't just selected in Africa, they were continuously rewritten as humans moved into new disease environments and encountered new pressures no previous generation had faced. Laure Segurel's team noted in their 2013 follow-up paper in Bioessays that the pressure maintaining this polymorphism for 20 million years likely also involves co-evolution with gut pathogens, microbes and parasites in the digestive tract that interact with the same sugar antigens displayed on red blood cells. The International Society of Blood Transfusion currently recognizes 48 blood group systems and more than 600 individual antigens on red blood cells. A, B, O is the most important for transfusion medicine. The rest are quietly doing something, interacting with pathogens, modulating clotting, shaping vulnerability and resistance in ways science is still cataloging. The full accounting of why we come in 4 types and exactly which diseases shaped each one is not yet complete. The next time you see your blood type written somewhere, on a donor card, in a medical record, on a hospital bracelet you didn't ask to be wearing, it is worth pausing for a moment on what it actually represents. Not a category, not a code, a record. 20 million years of plague, migration, and survival, written in sugar molecules on the surface of your cells, in the same molecular language that your gibbon cousins on the other side of the world are still carrying today. Your blood type is the oldest autobiography you'll never read.